Welcome. This discussion today is to replace the discussion in class if you may have missed any or part of it. As you go through these notes, you'll notice that your handouts are completely filled in. However, you will hear me talk about some information and use words and explain things that is not included in your notes. I would highly encourage that you take a moment to add this information to them. Also, at the end of your handout, there's going to be a series of questions that you would need to kind of answer as you go through this discussion here. We've seen this picture before. We built a model of skeletal muscle using the Q-tips and toilet paper rolls, construction paper, and the like. Once again, just to go through the macroscopic or large-scale anatomy of skeletal muscle, we have tendons that attach muscle to bone. The whole purpose of a tendon is to attach muscle to bone, and then the muscle itself begins right where we see this transition from white into red. So we have a question that kind of comes up: why are tendons white when they're attached to muscle, which is red? The answer to that: tendons happen to be avascular, whereas muscles are vascular, meaning that there is a blood supply. Or in tendons is not a blood supply. Surrounding the entire muscle itself is a layer of tissue known as the epimysium. This surrounds the entire muscle. Within the epimysium of the muscle, we can see that the skeletal muscle is highly compartmentalized. We see a bunch of these little strands. This one happens to be pulled out, but we see a bunch tucked in. The strand that kind of sticks out right here is known as a muscle fascicle, which is a collection of cells. So inside here we see one muscle cell, or a muscle fiber as they're otherwise known as. Surrounding each of these muscle fascicles is another little layer. We see it in white right here. That would be the perimysium that would surround each muscle fascicle. And then in here, surrounding each muscle fiber, we see more white stuff and other tissue known as the endomysium. So this is not new. We read about this stuff prior, but I just wanted to recap on it before we move forward. The next slide, we're going to be looking at the tip of this right here. So we're going to actually be going inside one of these muscle fibers. And when we get to it, we can see that it is also compartmentalized. So this is one muscle fiber right here, this entire thing. Once again, we're looking inside the tip of the end right here. So we're getting microscopic at this point. Inside, we can see that the compartmentalization units are known as myofibrils. Within one myofibril, we can see that there's a bunch of repeating patterns. These are known as sarcomeres. A sarcomere begins at a Z line and it ends at a Z line. And looking here, we can see where one sarcomere ends, a new one begins. And where this one ends, a new one begins. So these are stacked end to end going along skeletal muscle, and these fibers can be quite long. In some locations of our body, they can be upwards of 30 centimeters in length. That's a really, really long muscle cell. What we notice inside of each of these sarcomeres are two proteins: actin, which appear in purple here, and myosin, which appear kind of in, I guess, purple still, but more of a reddish purple color. Notice that myosin is thicker than actin, which is why sometimes we call myosin the thick filament, because it's thicker. Ultimately, what we have as a result of these sarcomeres and the presence or absence of, frankly, myosin, is striations. Striations are light and dark bands. Where we have the thick filament myosin, we tend to get a dark band, which we kind of see down here, represented by this deep dark color. And where there is no myosin, we have a lighter band. So the dark, light creates a banded, striated pattern within this muscle fiber. Also, before we move on, a couple things I wanted to point out because they will come up later. Also inside a muscle cell, we have a couple units: the sarcoplasmic reticulum, which is a kind of tubule system that kind of surrounds each myofibril, as well as a transverse tubule, which kind of surrounds each of the sarcoplasmic reticulums. Those two units will come up later on as we begin to look at muscle contractions. But first, let's talk about these striations. Once again, the light-dark bands caused by the presence of myosin, dark, no myosin, light. Here we see that definite striated pattern, the light and dark bands. Once again, a dark band represents where myosin is, and the light band represents where there is no myosin. Also, going back here, we can see that where there is no myosin, that's where the Z line exists. So we know that every time we see a light band here, we know that a sarcomere is beginning and ending there. Sarcomeres are quite small. What we see in this space right here is 1/10 of a millimeter. That is ridiculously small. If we were to count all these sarcomeres, I counted 17 when I was discussing this with class. That would mean that there's 170 sarcomeres based on this image in 1 millimeter, which is, exceptionally small of its own. The space between this right here is probably close to 2 millimeters. So there'd be, in reality, like 340 sarcomeres just in that space in actuality. Another thing I want to talk about in this image is that skeletal muscles do have more than one nucleus. Recall from your biology course that the nucleus, it controls cellular activities. Due to the sheer length of a muscle fiber, upwards of 30 centimeters, that's a lot of space for one nucleus to control. So there's going to be multiple nuclei, upwards of hundreds actually, that will help dictate what this skeletal muscle does, help keep it healthy, nutrients, repairs, so on and so forth. Here again we are just illustrating a couple things, again re-emphasizing that sarcomeres begin and end at a Z line, and they are stacked one after the next, the entire length of a myofibril. Again, we have some other substances that we're starting to see. We have the cell membrane of a muscle cell known as the sarcolemma. That term will come up. It surrounds the entire cell. It allows things to enter or exit a cell, predominantly is the function of the cell membrane. We can also see the striated patterns right here as well, the light and dark bands. Light bands representing once again that there's a Z line here and no myosin. Dark band would be where the myosin is located, and this would be the middle part of a sarcomere. Now, beginning to look at how muscles contract. Muscles contract in what is known as the sliding filament theory. It occurs in the sarcomeres, and we can see that here is a sarcomere that is relaxed, and down at the bottom we have a sarcomere that is contracted. Now, when a sarcomere contracts, we see the two proteins. The blue in this case is myosin. The red in this case would be the actin. When we go from a relaxed to a contracted state, notice that the length of actin here and contracted, they are the same length. So in essence, the length of actin, it does not get shorter when a muscle contracts. Neither does the myosin. We can see that the myosin down in the contracted state as well as the relaxed state are the same length. Those don't get shorter either. All that happens when a muscle contracts is that the Z lines move more inward, so to speak. In other words, the actin filament appears to slide across the myosin. Hence the term the sliding filament theory, not the shortening filament theory. They just slide and change position, causing the Z lines to move closer to one another. Ultimately, that's a contraction. Now, what we need to know about how these muscles are able to slide is, see these little fingers on myosin? Those are known as, right here, those are known as the myosin heads. We'll talk about the myosin heads shortly. But like fingers, fingers are meant to grab things, and myosin is the protein that actually grabs onto actin. It's not the other way around. Actin does not grab, myosin does. So those heads that I was just pointing to on myosin, they need something to grab onto. These little holes on these little bowling ball looking things look like a premier spot for those fingers to grab onto. Those holes are known as myosin binding sites. They're located on this entire unit, which is known as actin. So here we see actin, and here we see actin. Now what's different from the top image of actin to the bottom image of actin is up here those binding sites are not seen, and down here they're now visible. So what causes these holes to be covered up or revealed? Really, that's the answer to that would be an interaction between three things. A protein known as troponin, a protein known as tropomyosin, which is a rope looking thing, at least in this picture, and we can see that troponin is actually attached to tropomyosin. And then down here we see calcium ions playing a role as well. So let's talk about how this occurs. When there is no calcium in a sarcomere, this is what actin looks like. The binding sites are not closed, or are closed. That means that myosin cannot grab onto actin. A muscle in this state would be relaxed because the fingers of myosin have nothing to grab onto. However, when a nerve impulse releases calcium into a sarcomere, and we'll talk more about that later, the calcium goes directly to the troponin and it sticks to it. When calcium sticks to troponin, troponin causes tropomyosin, which is the protein that is covering up the holes, to shift out of position. When the protein tropomyosin shifts out of position, these holes are revealed. Once that occurs, myosin will grab onto actin. And when myosin grabs onto actin, we form what is known a cross bridge. Here we see a cross bridge. We have the head of myosin, which is now able to grip onto actin, which is this entire top thing. Unfortunately, this particular image does not show the binding sites revealed, but since the myosin is coming into contact with actin up here, it's implied that they are. This will conclude the first video of this section. The second one will pick up where this one left off and talk about how a muscle actually contracts.